When we think of infectious diseases, bacteria and viruses usually come to mind first. But beyond these familiar threats lies another vast and largely hidden kingdom, the fungi. In this panel, we meet a curious medical student peering through a microscope, about to discover that fungi are far more than just the mushrooms we see in the forest. They are a diverse group of organisms with profound importance in medicine, ecology, and everyday life. Unlike bacteria, which are simple prokaryotic cells, fungi are eukaryotes, meaning their cells contain a true membrane-bound nucleus along with a full complement of complex organelles, including mitochondria and endoplasmic reticulum. This places fungi in the same cellular category as human cells, which is precisely what makes treating fungal infections so challenging. Any drug that targets a fungal cell must be selective enough to spare the structurally similar human host cells. Here we see one of the most clinically important differences between human and fungal cells, the composition of their cell membranes. Human cell membranes are stabilized by cholesterol, while fungal cell membranes rely on a structurally distinct sterol called ergosterol. This single biochemical difference is the primary target of several major classes of antifungal drugs. By disrupting ergosterol synthesis or binding to it directly, antifungal agents can selectively damage the fungal membrane while leaving human cells largely unharmed. Another feature that sharply distinguishes fungi from human cells is the presence of a rigid cell wall. This wall is composed of three main layers. The innermost layer consists of chitin, a tough, fibrous polysaccharide also found in the shells of insects. The middle layer is made of glucan, a complex sugar polymer that provides structural integrity. The outermost layer contains glycoproteins, which are sugar-coated proteins involved in cell signaling and adhesion. Because human cells lack a cell wall entirely, these structural components represent attractive and selective targets for antifungal drug development. The vast majority of fungal species are saprotrophic organisms that live freely in the environment, particularly in soil and water, where they feed on dead and decaying organic matter. This role is ecologically indispensable. Fungi are nature's primary decomposers, breaking down complex organic compounds and recycling nutrients back into the ecosystem. Without fungi, dead plant and animal material would accumulate indefinitely, and the nutrient cycles that sustain all life on Earth would grind to a halt. Not all fungi are harmful. In fact, many are profoundly beneficial to humanity. Yeast, a single-celled fungus, has been used for thousands of years in bread fermentation and the production of alcoholic beverages. Certain molds, particularly species of penicillium, are essential to the production of aged cheeses. And perhaps most famously, Alexander Fleming's discovery of penicillin from the mold Penicillium notatum in 1928 launched the antibiotic era, saving hundreds of millions of lives. Fungi, it turns out, are among our most valuable biological allies. Not every fungus that lives on or in the human body is a threat. Many fungal species, most notably Candida albicans, exist as commensals, organisms that live harmlessly alongside their host, neither causing harm nor providing direct benefit. Candida is a normal resident of the human skin, mouth, gastrointestinal tract, and genitourinary system. Under ordinary circumstances, it coexists peacefully with the host's immune defenses and the surrounding microbial community, causing no disease whatsoever. The story changes dramatically, however, when the host's immune system is compromised. In patients receiving chemotherapy, immunosuppressive drugs, or those living with HIV, the delicate balance that keeps commensal fungi in check can collapse. Candida and other opportunistic fungi seize this opportunity, proliferating rapidly and invading tissues they would never normally penetrate. This transformation from harmless resident to dangerous pathogen is the hallmark of opportunistic mycosis, infections that exploit vulnerability rather than inherent virulence. Fungi can be broadly categorized by their physical form. Filamentous fungi, commonly known as molds, grow as long, branching, tube-like structures called hyphae. These microscopic filaments extend outward in all directions, penetrating and colonizing their substrate. Hyphae may be septate, divided by cross walls called septa or cenocytic, meaning they lack internal divisions and contain multiple nuclei in a continuous cytoplasm. The type of hypha is an important feature used in laboratory identification. As hyphae grow and branch repeatedly, they interweave to form a dense, three-dimensional network known as the mycelium. The mycelium is the main body of a mold, and it is responsible for nutrient absorption, structural support, and ultimately reproduction. What we see as a fuzzy patch of mold on bread or fruit is the visible surface of a mycelium that may extend much deeper into the substrate. In nature, mycelia can spread across vast areas underground, forming some of the largest living organisms on Earth. In contrast to molds, yeasts are unicellular fungi, single, oval-shaped cells that reproduce primarily through a process called budding. In budding, a small outgrowth forms on the surface of the parent cell, gradually enlarges, and eventually separates to become a new independent cell. Two medically important yeasts are Candida albicans, the most common cause of fungal infections in humans, and Cryptococcus neoformans, which is distinguished by its prominent polysaccharide capsule, a key virulence factor that helps it evade the immune system. Some of the most dangerous fungal pathogens are dimorphic, meaning they can exist in two distinct morphological forms depending on environmental conditions. At environmental temperatures around 25 degrees Celsius, these fungi grow as molds, producing hyphae and spores in the soil. When they infect a warm-blooded host and encounter body temperature of 37 degrees Celsius, they switch to a yeast form. This thermal dimorphism is a critical virulence mechanism. A classic example is Histoplasma capsulatum, the causative agent of histoplasmosis, a potentially serious respiratory infection. Fungi reproduce primarily through spores, microscopic reproductive units that can be produced in enormous quantities. Spores may be formed sexually or asexually, and they are released into the surrounding environment in vast clouds. A single fungal colony can release millions of spores in a single day. These spores are extraordinarily resilient, capable of surviving harsh environmental conditions including desiccation, ultraviolet radiation, and extremes of temperature, allowing fungi to colonize nearly every habitat on Earth. The primary route of human exposure to pathogenic fungi is inhalation. Fungal spores are small enough to remain suspended in the air for extended periods, and when inhaled, they can travel deep into the respiratory tract, reaching the alveoli of the lungs. For most healthy individuals, the immune system clears these spores without incident. However, in immunocompromised patients, or when the inhaled spore burden is very high, pulmonary fungal infection can take hold, sometimes with severe consequences. This is the initiating event in many systemic mycoses. In the clinical microbiology laboratory, the morphological characteristics of spores are among the most important features used to identify fungal species. The size, shape, color, surface texture, and arrangement of spores, as well as the structures that bear them, provide a detailed fingerprint that allows trained mycologists to distinguish one species from another. This morphological identification is complemented today by molecular techniques, but microscopic examination of spore characteristics remains a foundational skill in diagnostic mycology. Fungal infections are classified according to the depth of tissue invasion. The most superficial category involves infections confined entirely to the outermost layer of the skin, the stratum corneum, as well as the surface of hair shafts and nails. These superficial mycoses cause minimal or no inflammatory response, as the fungi do not penetrate living tissue. Examples include tinea versicolor, caused by Malassezia species, and black piedra. While not life-threatening, these infections can cause significant cosmetic concern. Cutaneous mycoses go one step deeper, involving the keratinized layers of the skin, hair, and nails. The causative organisms, collectively called dermatophytes, possess the ability to digest keratin, the structural protein that makes up these tissues. Common dermatophyte infections include tinea capitis, affecting the scalp, tinea corporis or ringworm of the body, tinea pedis, commonly known as athlete's foot, and onychomycosis, a fungal infection of the nails. These infections are among the most prevalent fungal diseases worldwide, affecting hundreds of millions of people. Subcutaneous mycoses involve the deeper layers of the skin, subcutaneous tissue, and sometimes underlying muscle and bone. These infections typically arise from traumatic inoculation, when fungal spores or hyphal fragments are introduced directly into the skin through a puncture wound, often from a thorn, splinter, or soil-contaminated injury. The infection then spreads slowly through the subcutaneous tissue. A classic example is sporotrichosis, caused by Sporothrix skanki, which is sometimes called rose thorn disease because of its common mode of transmission. Systemic mycoses represent the most severe category of fungal infection. These infections typically begin in the lungs following inhalation of spores, and in susceptible individuals they can disseminate through the bloodstream to virtually any organ in the body, including the brain, liver, kidneys, and bones. Systemic mycoses caused by primary pathogens such as histoplasma, coccidioids, and blastomyces can affect even immunocompetent individuals, while opportunistic pathogens like Aspergillus and Cryptococcus predominantly target the immunocompromised. Beyond direct infection, fungi can harm human health through allergic mechanisms. Fungal spores and hyphal fragments are potent aeroallergens, capable of triggering IgE-mediated hypersensitivity reactions in susceptible individuals. Chronic exposure to airborne mold spores is a well-established risk factor for the development and exacerbation of asthma, allergic rhinitis, and allergic fungal sinusitis. Certain mold genera, including Alternaria, Cladosporium, Aspergillus, and Penicillium, are among the most clinically significant sources of fungal aeroallergens in both indoor and outdoor environments. Some fungi produce highly toxic secondary metabolites known as mycotoxins, which can contaminate food crops and pose serious health risks when ingested. Among the most dangerous are aflatoxins, produced by Aspergillus flavus and Aspergillus parasiticus, which commonly contaminate corn, peanuts, cottonseed, and tree nuts, particularly in warm, humid storage conditions. Aflatoxins are among the most potent naturally occurring carcinogens known, and their presence in the food supply is a major public health concern in many parts of the world. The link between aflatoxin exposure and liver cancer is one of the most thoroughly documented examples of a mycotoxin causing human malignancy. Aflatoxin B1, the most toxic and carcinogenic of the aflatoxins, is metabolized in the liver to a reactive epoxide intermediate that binds covalently to DNA, causing mutations in the tumor suppressor gene TP53. Chronic dietary exposure to aflatoxin is strongly associated with an elevated risk of hepatocellular carcinoma, primary liver cancer, particularly in regions where hepatitis B infection is also prevalent, as the two risk factors act synergistically. The development of effective antifungal drugs is one of the great challenges of infectious disease medicine. Because fungal cells are eukaryotic and share many fundamental biological processes with human cells, finding drug targets that are present in fungi but absent in the host is difficult. The ideal antifungal agent exploits the unique features of fungal biology, such as ergosterol in the cell membrane and chitin and glucan in the cell wall, to achieve selective toxicity, eliminating the pathogen while causing minimal harm to the patient. Antifungal resistance is an increasingly serious clinical problem, and fungi have evolved multiple mechanisms to evade drug action. One of the most common is the overexpression of efflux pump proteins, membrane-spanning transporters that actively expel drug molecules from the fungal cell before they can reach their intracellular targets. This mechanism is particularly important in azole resistance among Candida species. By continuously pumping the drug out of the cell, the fungus maintains sublethal intracellular drug concentrations, allowing it to survive and proliferate despite treatment. A second major resistance mechanism involves mutations in the gene encoding the drug's molecular target. In the case of azole antifungals, which work by inhibiting the enzyme lanosterol 14-alpha demethylase, encoded by the ERG11 gene, point mutations can alter the shape of the enzyme's active site, reducing the drug's binding affinity without significantly impairing the enzyme's normal function. The result is a fungus that continues to synthesize ergosterol normally but is no longer susceptible to the drug. Such target site mutations have been identified in resistant clinical isolates of Candida albicans and Aspergillus fumigatus. A third resistance strategy involves the activation of alternative metabolic pathways that allow the fungus to circumvent the blocked step entirely. When a drug inhibits a key enzyme in a biosynthetic pathway, some fungi can upregulate alternative enzymes or pathways that produce the same end product by a different route, effectively bypassing the drug's point of action. This type of metabolic flexibility represents a significant challenge for antifungal drug development, as it means that even highly potent inhibitors of a specific target may be rendered ineffective if the organism can route around the blockade. Fungi are everywhere. They inhabit the soil beneath our feet, the air we breathe, the food on our tables, and the surfaces of our own bodies. They are essential decomposers, industrial workhorses, and the source of life-saving medicines. Yet they are also capable of causing disease, from the superficial and cosmetic to the systemic and life-threatening, particularly as the global population of immunocompromised individuals continues to grow. Understanding the biology of fungi, their unique cell structures, their modes of reproduction, their mechanisms of pathogenesis, and the ways they resist our drugs is not merely an academic exercise. It is the essential foundation for diagnosing, treating, and ultimately preventing the full spectrum of mycotic diseases that affect millions of people around the world every year.